I assume this is a mid-20s woman who likes to party a lot. These are windows, of course. I doubt she knows the exact time, but if you were in the liquor store, then you should.
2 hours.
And then here's the one I like. So Kings of Leon concert, and then overnight, 1 a.m. to 3 a.m. Best Western. What was she doing in Best Western for 5 minutes? I don't know. Don't want to know. Anyway, just thought it was amusing. I put the link up to the article from NPR. This one on Facebook. You can check that out. Okay, so we'll delve into chapter 23, viruses and cancer. Let's start with a definition of cancer. Somebody define cancer. Zodiac sign, graph.
Uncontrolled cell growth.
I'm sorry?
Uncontrolled cell growth.
Just so right, but yet unentertaining. Right, it's uncontrolled cell growth. So, a more strict definition, and one will come into contact with in the paper today. Is transformation from senescence. So, in developmental biology pathways, you have stem cells that become tissues and lead to cells that no longer grow and divide. Those are senescent cells. So something happens in senescent cells. For example, infection with a virus that turns on a gene that leads to a loss of control of the cell cycle and pushes the cell into out of G into S. You lose senescence and become tumorous. Now, of course, all cancers are not caused by, all cancer is not caused by viruses and radiation, environmental factors, and viruses are also included in that. Normal genes, so for example, BRCA, it's a breast cancer gene. If you're positive for an allele of BRCA1, I believe, you're more or less going to get breast cancer. This is anyway, there's screening for people with that allele, but they're normal alleles that cause cancer processes later in life. But we're, of course, concerned about viruses. Viruses that cause cancer are oncogenic viruses.
Is that an L?
It's a 1.
Oh, 1.
Just naming that as an example. There's genetic predisposition to cancers. And approximately 20% of cancers in females, 8% in males are attributable to viral infections. Taking a quick tour through the parade of viral cancers. We start with papillomavirus that leads to cervical carcinoma. So your book is showing cancers in females and males. What percentage is attributable to viruses? You see cervical cancer by far the highest. This is a weird number to me because I was of the knowledge that it was substantially more than that. So I believe actually the paper, the number 80% is popping into my head. But anyway, a significant percentage of cervical carcinomas are caused by HPV. As we'll see in a minute, or in a few minutes, we'll talk about the biology of that in a bit. 500,000 cases per year, 275,000 deaths. HPV must enter through abrasions. Epidermal cells in mucous membranes, and thus it's spread by genital contact and is, of course, an STD. It infects both males and females, but leads to cervical carcinoma only in those of us with cervixes. Actually, a high school classmate of mine who's a faculty member at the University of Minnesota was just granted the Distinguished Teaching Award for teaching a course about the evolution of sex. And she has lab sections where they talk about anatomy using sex toys, so be happy I'm not that innovative. And it's in persistent infections, so persistent infections lead to the carcinogenesis. You could get HPV and then clear the infection, so we're talking about a small fraction of HPV infections leading to cancer, but it's a significant percentage of the cancer caused by these persistent infections. There are a number of strains of HPV. I just wanted to point out on the phylogenetic tree, the ones we'll be talking about later are the high-risk strains, which are 18 and 16. So they're phylogenetically distinct. Again, these are very minor differences compared to, say, elephants and E. coli. But there are strains known to, actually the ones in the orange thing, they're known to lead to cancers much more frequently than other strains. Polyoma virus linked, polyoma meaning many tumors. Include Merkel cell carcinomas and an example of a polyoma virus is SV40, simian virus 40, which I bring up not only because it's in your textbook, but is and we'll see an example of this in the paper, a commonly used viral promoter in modern research. So if you want to transfect a mammalian cell with a gene that's going to express a lot of copies of the protein, you'll drive it with an SV40 promoter. So it's a very strong promoter. An example of a virological event leading to a research tool. It was originally isolated from primary monkey cultures. I'm sorry, is there something wrong? I'm hearing a murmuring. That were used to produce the polio vaccine. So a historical example actually of people getting viral infections and eventual cancers from a vaccine developed. This is one reason why we don't do them in monkey cells anymore. So maybe those crazy anti-vaccine people were onto something. In the 1960s, perhaps, you could get cancer from a vaccine. Of course, you can read this in your book, at least for purposes of the next 5 or 4 weeks. Epstein-Barr virus is associated with Burkitt's lymphoma. So c-Myc is a oncogene expressed abnormally high levels. In cancer cells, Epstein-Barr facilitates an increase in c-Myc, and then this affects B cells. Burkitt's lymphoma is a B cell cancer. Kaposi's sarcoma, very frequently found in people suffering from AIDS, is caused by a herpes virus that we saw earlier in the course, KSHV. Yes.
What type of gene did you say c-Myc was?
I'm sorry, so c-Myc is an oncogene. I bring it up, and I forgot to tell you why I brought it up. For one reason, not particularly the Epstein-Barr. These are very important things, of course, but relative to an undergraduate survey course, what I'd like for you to, well, you'll see c-Myc in the future as you do see SARC, primarily because it's become an excellent a research tool as well. So we put MYC tags, a little piece of the protein on proteins we're interested in, which makes them very antigenic. So this is an example of a gene that is overexpressed, leading to driving the cell in from G into S. We're going to see a research example of this with a paper later, not with c-Myc, but with some other proteins. So most of these viral cancers are caused in large measure by something that the virus does, either a viral gene or an upregulation of a cellular gene that causes the cell to lose control of the cell cycle. So I'm jumping ahead to my example of HPV. Knocking out the p53 tumor suppression pathway, thus leading to proliferation. So abnormal levels of proteins that control the cell cycle. c-Myc is one, leads to cancer, and that's in the Burkitt's lymphoma. T-cell leukemia can be caused by, this is an easy one to remember, human T-cell leukemia virus 1. That's a retrovirus. And then the leading cancer in men, hepatocellular carcinoma, associated with Hep B and Hep C viruses. This is the only known plus RNA virus to be oncogenic.
What's after T-cell leukemia?
HTLV, human T-cell leukemia virus 1. So why might you not expect a plus strand RNA to be oncogenic?
Never, doesn't necessarily need to enter the nucleus.
Yes, there are plus strand RNAs that do. I was thinking that would be a good final exam question. It's that easy to answer. Maybe it still is. Right? They're not going to be viruses that require the genome to enter the nucleus, and therefore you're not going to have rearrangement with the host chromosome. Therefore, you're not going to have changes in host gene expression induced by the virus. Except in hepatocellular carcinoma, and it's that the oncogenicity of the virus is likely because it causes cirrhosis, like, you know, 2 R's. Never remember whether cirrhosis has 2 R's or 2 H's. Cirrhosis leads to a regrowth of, or an attempt by the liver at least to regrow functional cells, which leads to defects in cell division. We've been trying to do it frequently. Defects in cell division that do lead to cancer. So it's a different way of generating a cancer than integration into the chromosome with the retrovirus, for example. Yes, Priscilla.
I'm so sorry. Can you repeat why positive strand RNA viruses can be?
So what is a positive strand of RNA. What's the difference between positive and negative strand? The positive is the same as an mRNA, and so a positive strand virus can infect, have its RNA in the cytoplasm where it's ready to be translated into viral proteins. It doesn't go into the nucleus as minus strand or DNA viruses or retroviruses do as part of their replication cycle. Because it's not in the nucleus, it's not near the host chromosome. The retrovirus, for example, does integrate into the host chromosome. It says integrations into the chromosome that result in changes in host gene expression that can lead to oncogenesis. So you would not offhand expect a plus-strand RNA virus to be oncogenic. This one tends to be Hep B and Hep C tend to have a role in causing a hepatocellular carcinoma because they damage the liver, and it's the body's attempt to replace that or to fix that damage that leads to oncogenesis. So that's much better. All right.
Just real quick.
Please.
The hepatocellular carcinoma, you said it causes cirrhosis?
No,
Then
the
leads-
virus causes cirrhosis. So if you have Hep B infection, there's inflammation, death of liver cells. If you attempt to replace those dead cells, the cirrhosis is scar tissue in the liver from cell death. So they attempt to replace those with newly derived hepatocytes because you have to regrow, so you have to do cell division. Mistakes in the cell division that can generate cancer. All right. So medical privacy wasn't always what it is today. And I know Brandon's looking for a good book now that he's finished The Great Influenza. So I'm going to recommend, if you haven't read this, go read The Immortal Life of Henrietta Lacks. It came out a couple of years ago. Fascinating book. If you want to go to med school, you're going to want to be able to talk about it in your interview because all the doctors have read it because I think it's so freaking cool. And it is. It's an awesome book. This is the part of the class where we're going to talk about virus, tissue, cell lines derived from virus-associated cancers. So for 60 some odd years now, the strain of cells known as HeLa cells, it's a cervical carcinoma line, have been widely used in research. It was the first cell line that was ever immortalized or grown in culture in an immortal way. So one of the problems with cell culture is if you don't have an immortal line, i.e. cancer cell line, if you're growing primary cells, like those primary monkey cells, you can take cells out of an organism, put them in a plate, and they grow and divide until they stop because they become senescent. So primary cells are bad for a lot of research purposes because they stop growing after a while. The first human line, there were some chicken lines and other things that were developed, was grown by a guy at Johns Hopkins. His name was Gay. And they were isolated from a patient who died in 1951 of cervical cancer. So for years and years and years, he had produced this cell line, distributed all around the world, and it was called HeLa. It turns out that he named it HeLa because the patient from whom the cells were isolated was named Henrietta Lacks. And this is how bad medical privacy was. He named the cell line that he took without her knowledge from her cervix after her. So I just wanted to do a little of the history here because this does bring up some interesting bioethical issues. It's a neat story. And it's an excellent example of a research strain derived from virally induced cancers. The woman named Henrietta Lacks, born in 1920, died at age 31 in 1951. She was from rural Virginia. Her family were tobacco farmers. She married her first cousin, which is not a rare event. Actually, still in many places today. I have several first cousins in my family tree. Reason why I am the way I am. She had 5 children. She started feeling a knot in her stomach in early 1951. Her family goes, oh, you're just pregnant again. It turns out she was pregnant. Right after she delivered her 5th child, she had uncontrolled bleeding, lots of pain, went back into the hospital, and her family had moved from rural Virginia to Maryland to work in, I believe, steel mills along with her husband. So Johns Hopkins was one of the only major medical centers that would treat African American people at that time. And so just by happenstance, she happened to be one of the best hospitals in the country. Unfortunately, there was no decent treatment for cervical cancer at that time. She did get the entirely appropriate standard of care for the time, which were having tubes of radium sewn into her cervix. So it was an early radiological treatment. And so she got that treatment. Of course, it did a lot of damage. Went back into the hospital where she was admitted, given radium tubes again, and then died. But during her treatments, the doctor who did biopsies of her tumors and gave them to the pathologist to actually that's how they diagnosed the epidermoid carcinoma, was the pathologist at the hospital, did the lab test, did the pathology and said, oh, this is this. They also noticed that they just kept on growing. So this is one of the most aggressively growing sets of cells ever seen. It was, again, the first human cell line to be immortalized. He named it Gila. She died of total kidney failure because her cancer had metastasized all over her body, very quick from diagnosis to death, as was very common at that day. And it was the aggressiveness of the cancer cells that led to their immortalization as a HeLa cell. One of the ways senescence works is by, on your chromosome, at the end of your chromosomes, of course, you have things called telomeres. So it's non-coding sequence. It's thought to essentially be a molecular clock. And as you get older, your telomeres get shorter. And then eventually you lose your telomeres, and this is one of the processes of aging. Things start to break down. An enzyme called telomerase is involved in cell division, and it synthesizes telomeres. One of the things often seen in cancers is an upregulation, an inappropriate one, of telomerase, which allows for continual division of cells. And so in HeLa cells, there is a very high level of telomerase activity. One of those things you might remember in a few years. A Hayflick limit. It's the number of times a cell culture, or a number of divisions in order to, can divide. So if you're growing primary cells, maybe you get 8 cell divisions before they stop growing. HeLa cells have multiple infections with HPV strain 18. If you read the book, incidentally, you'll discover that there's ample evidence that her husband/cousin was unfaithful to her on a number of occasions. It was almost surely he who infected her with HPV. There's no evidence, which is not well known, of course, at the time during her life. There's a cautionary tale there, I'm sure, about who you marry. But this infection with HPV-18 generated, or led to the cancer, for ways we'll show in a minute when we get to the paper. But it turned off, so the HPV produces 2 proteins that turn off tumor suppressor pathways, and it was an integration of HPV into her cervical cells that led to the cancer. She also, he was also probably responsible for infecting her with a number of other venereal diseases that were discovered, and that weakened her immune system and perhaps made things worse faster. So, here's the guy who grew her cells, and the bioethics here is that she was never told that her cells were being taken for research purposes. Her family was never told. Her family was largely, even to this day, very poor, uneducated, don't understand what happened, and well into the 2000s until Rebecca Skloot started writing the book and talking to the family, we're not really sure whether their mother was dead or not, because they had learned after these cells became widely used, geneticists started knocking on their doors going, hey, can we take some samples from you because we want to figure out what's going on in these HeLa cells. Because in one way, their family members' cells are still growing. It is so widely used, 50 million metric tons of those cells have been grown around. Immense scientific benefits. First tested the polio vaccine used in cancer, AIDS research, development of IVF, all sorts of cloning technologies have been developed using these HeLa cells. Since the development of HeLa cells, there are now dozens, hundreds, thousands of carcinoma lines for various different types of cells. But this is sort of the workhorse of eukaryotic biology as E. coli is for microbiology. It's so aggressive, incidentally, it's also caused a lot of damage in the research community because they're so good at growing, even a slight amount of contamination leads to them overgrowing other cell lines. There have been people thinking they're working with, hepatocyte cell culture, but really it's HeLa cells that overgrew culture 20 years ago, and it's wrecked a number of dissertations. And then there are commercial firms that produce and sell HeLa cells. The Tuskegee Institute actually was the first company, is not the right word because they're an institute, first group that started manufacturing HeLa cells on a commercial basis. So there's some other interesting questions we won't get into, which is, should her family get any money from the products of her cells? Her family is now aware of what happened, and there are people trying to raise money. There's a Henrietta Lacks Foundation that's trying to raise money to help her family and other people who've had this happen to them. But on the other hand, no damage was done to her. She got the highest standard of care for cervical carcinoma that existed in 1951. So there's the counter argument is, having taken her cells, it didn't damage her at all. It did a lot of good. So there's a bioethics issue there you can go either way on. Courts have decided, by the way, that you don't own your cells. So this was 3 or 4 years ago now. There was a court case that if you're interested, I can look up and talk about it for a little more material. So every one of us, at least any of us who've ever gone to a doctor and had clinical samples taken, have had our tissue, after the test is done, deposited in tissue sample databases is the wrong word, but there are warehouses full of tissue samples that include your samples, and scientists will pull them out and test them as they want. Most of them, of course, will never be used, but there are high throughput search strategies going on. And there was a case, I think it was in California, where a guy sued, and the court said, well, you don't own this because you gave it up freely as part of diagnostics. So they still have you sign a form that says you give it up, but you have no right to your own cells, which is discussed in the book. So it's an interesting and a little bit scary thing. It's redounded to Henrietta Lacks's family because last year somebody finally published her genome, the HeLa genome. And so that's putting out, if you're a geneticist, you can figure out the likelihood of her descendants having particular alleles. So there's a medical privacy issue there as well. And we'll see HeLa cells in today's paper. I've already said all this stuff. Let me just read. So viruses cause cancer. By usually persistent infection. Why is one acute infection probably not enough?
Use that to make them,
That's like the resonance of virology, yes.
it's not enough to improve the cells to become resonant.
Yes, but it's because these events that cause cancer are very rare. Your virus that integrates into the genome, randomly speaking, it's not very frequently going to muck up control of a cell cycle protein. So multiple copies of HPV-18 in HeLa cells, why? It's because it took a long time for that virus to land in a spot where it became oncogenic. So persistent infection, because the viral events that lead to cancer are rare events. So that also tails into a small percentage of the infected develop cancer. Because it's a rare event. Often not viruses alone. For example, Henrietta Lacks had multiple STDs. It may have suppressed her immune system to the point where the cervical carcinoma escaped immune defenses, speaking of which. Immunodeficient individuals, environmental factors also, of course, play a role. So, as an example here, AIDS sufferers getting Kaposi's sarcoma. Kaposi's is very, very rare among people who do not have immunodeficiency. And so it's not just, oh no, I got measles from the Kings of Leon concert, I'm going to get cancer. Complex interplay of factors. And so, by the way, so I hope all of you saw, I switched the paper I'm going to talk about today because I got to reading and preparing for this class. I thought, oh, I should go get this paper because it's a really cool paper. And then I finished preparing my notes and I picked the right paper. So right at the end of your text, at the end of the chapter, increasing evidence that virus proteins synthesized during persistent infections play roles in conversion of normal cells to cancer cells. And that's what we're going to talk about. This paper.
I have a quick question.
Shoot.
If the HPV infection can cause cervical carcinoma, but you can have an HPV infection in both males and females, does it not cause cancer in males because they don't have a cervix?
It is, I don't know that it does not cause cancer in males, but it's the anatomy and biology of the cervix and the nature of sex itself that make females more susceptible to papilloma virus infections. And HPV will tend to be more persistent in women than men. That's at least one of the factors. So, coming back to the last two figures from the text, what we see is a typical retrovirus. One of the ways it can cause cancer by altering expression of a host gene in two ways. One of which is these LTR repeats that allow it to integrate into the chromosome and also serve, at least the 5' LTR, serves as a promoter for expression of the viral genes. Well, there's also, since it's a repeat, this is the same sequence down here, and so there's a promoter that's driving nothing as far as the virus is concerned, but if it integrates into the host cell upstream of a cellular gene, it can cause inappropriate expression of that gene, leading to a loss of, if that's the wrong gene, leading to a loss of control of the cell cycle. It can also, in recombining and emerging from the chromosome to produce progeny, adopt a host gene as part of the viral genome. You see that with the Sark genes and some other examples, and thus the viral infection itself and transcription of the viral genes can lead to expression of the cellular, of the host protein via the viral genome. Relative to HPV, oops, sorry, this is not HPV, this is HTLV. It is an example of an auxiliary protein from the virus serves as in combination with host cells. That auxiliary protein, one binds to a cell cycle regulatory protein leading to loss of cell cycle, and also bound to a host protein is a transcription factor for the viral genes, but will also drive cellular genes. So an example of a viral gene expression causing loss of cell cycle control of the host. HPV, we'll see in a minute, the E6 and E7 genes encode, of course, E6 and E7 proteins, which bind to P53 and PRB, which has a name, I'll get to in a minute. These are these are tumor suppressor proteins. We'll see what they do in a minute, but they arrest the cell cycle in G phase, and when they're expressed and active, they arrest the cell cycle. When these E6, E7 proteins from HPV bind to them, those tumor suppression pathways are lost, leading to cancer. And so this is part of the biology of the virus. You have an integrated provirus within the host. It causes cell division and growth of the host with the integrated provirus. So this is one of the ways the virus has of replicating. Which brings us to today's paper. I was reminded of this because when I was finishing up my PhD, you get to a point where you've been in school for a really long time, and you get graduation, and eventually you're not in school anymore, and there's this terrifying prospect that you have to get a job. And so I was 31 years old. It was a good run, right? I avoided any serious work. Actually, I still avoid any serious work. But I was finishing up at the University of Connecticut. Probably at that point, fiance was at Yale doing her residency, so I needed to get a job. Anyway, I wanted to get a job at Yale, so I started looking up labs and contacting them and say, hey, I just got a PhD, and a postdoctoral position for me. So I interviewed in this lab, and they didn't hire me. I'm still mad about that. But on the plus side, I did not become a virologist because of that, so maybe I should be happy about it. So this guy, Dan DeMeo, interviewed me, and I met for a long time with the first author, and she showed me this work, which had not been published at the time, so this was in 2002, this is a 2003 paper. And I was just reminded of this, because we're thinking cancer and viruses, and they had some really cool results. And I remember I was so thrilled with the process that these interviews lasted an entire day. And they always, 5 o'clock, would go meet with the boss one last time. And I was so thrilled by this one. This is awesome, sign me up. He's like, yeah, I don't think so. Turned out my graduate advisor had screwed me in my letter of reference. So that's why I'll never write anybody a bad letter. I will refuse to write letters, but I will not say bad things about people. Letters of reference, because anyway, we'll skip the abstract, I guess. Interestingly, I stalked her on the internet yesterday. She's still a postdoc 11 years later, so maybe it was a good thing I didn't get hired in that. She's now out in California somewhere doing something else. But, So this paper will deal with the high-risk strains HPV 16 and 18. The important thing to note now is that one, HPV 18, these are going to be HeLa cells. HPV 18 is in the cell line, it's integrated in the chromosome, and they're going to use 16 genes brought in exogenously. They can tell the difference between the two. It's very important. So there's some specific differences there. We're going to work with E6 and E7. Those are the ones from the figure. They are the major viral oncogenes for reasons we just talked about. E6 binds to P53. Interestingly, P50, so you'll very frequently run across proteins that have very bad names. Tumor suppressor proteins are named this way. So P53, does anybody know why it's P53?
Like where the gene is on the chromosome?
Nope. Good guess. Frequently named for that. Oh, P and Q, yeah. The P stands for protein. 53 what?
53 proteins
Nope.
discovered.
Also good idea. Also done. Not here.
53rd one guessed.
Nope.
So how many nucleotides?
53.
So it's a protein. It's amino acids. It's
53 amino acids, or 53.
no, 53 kilodaltons.
Oh.
So that's so somebody ran. So the first person to work with it ran out on a gel, and it ran in an SDS gel as something that weighed 53, or whose molecular weight was 53,000 grams per mole. It's a bad name for a number of reasons. One, there are other proteins that are about that size, and two, it's not actually that size. After it was cloned, it was discovered it's actually 43,000 kilodaltons. But the name stuck. It's 53. It's a tumor suppressor gene. We'll get into what it does. It does a lot of different things. But this binding event, A plus B going to AB, leads to ubiquitination of P53, followed by degradation. So the virus expresses its protein, it binds up that tumor suppressor protein, and is targeted for destruction by the cell. So E6 being there means P53 is eradicated. E7 binds P105, which is PRB used in the text. It's another tumor suppressor gene. We'll see more about what it does in a minute. The other protein here is E2. E2 negatively regulates E6 and 7 transcription.
I see.
What does that say out here in the second?
Transcription.
No, on the top of it.
P105.
P105
Oh, what's the next one?
equals PRB, so that's the tumor.
Okay.
It's this one.
Oh.
Textbook. So P53 is P53, PRB is P105. We believe they actually, they refer to it as 105 RB. So figure 1 is a northern blot. What's a northern blot?
It's an RNA.
Right, they're looking at RNA. Southern blot is DNA, western blot is protein. Northern blot of HeLa cells with the endogenous strain 18, 6 and 7 proteins. So this is one messenger RNA. And this is, so they're infecting it with a modified virus that provides a copy of these RNAs that is then that is in either, so HPV, so this is the endogenous E6 and E7, and then provided with E6 or E7 from a constitutive promoter. So, by the way, I put this paper out on D2L and said, oh, this is going to be an awesome paper, and but crap, this is really complicated. So, it doesn't work. So, E6 and E7 from the HPV integrated copy. And then these are integrated copy plus an exogenously provided copy of E6 from a different strain. So we're either looking at a probe on the blot to the 18 strain version of 6 and 7, or a probe on the blot to the 16 strains. We can tell the difference. And then the minus and plus. So these numbers are clones. So they're looking at two different samples for each condition. This is just the vector alone. So this is the E6 gene in this vector here. This is just the vector alone, so it's a negative control. And then the minus and plus is infection with the virus carrying the E2 gene. So let's start here. Here we have, and then the minus is mock infected, so they do everything except add the virus. So we have expression of E6 and E7, but when E2 is provided, we shut down expression of 6 and 7 because E2 is there. So this is negative regulation of E6, 7 transcription.
Okay,
Yes.
so go back to what you said about the E2. You said about the plus or minus.
Okay.
E2, if it's E2 is the positive one, right? If it's infected with E2, it's positive.
Yes. So, yes, the plus is infection with the virus that delivers a copy of E2. So when the E2 protein is made, E6-7 transcription is shut down, which is why there's nothing there. If it's infected with, so mock infection is whatever buffer the virus is in, they put in as well. That's, you can say, not infected. And it happens in both of them. So with a copy of E6 provided, the 18 expression is shut down. So same as this. And here's a copy of E7 provided, 18, E6 and 7 shut down.
So the E6 and E7 are coming from a different cell, or?
We're not there yet.
Okay.
So there are two blots here, this blot and this blot.
Right.
With this, so the way you do a northern blot is you run an RNA gel out and you transfer it over to a membrane. So you have total cellular RNA bound to a membrane. So once it's there, it doesn't come off. And then you take a radioactive or some other label, complementary sequence in a solution, you put it in, you shake it in a bag, and then you wash, and then you develop. So the normal way this is done is with radio labeled. So you have phosphorus 32, that's probably what they were doing here. This is back 2000. Now they have all sorts of fancy dyes that you shoot with lasers that make different colors. But this is black and white. This is the older days. And so with that, whatever that probe is, will hybridize, because it's complementary, to that one band on RNA, and then you put a piece of film over it. If it's the radioactive particles hit the film, create a black spot. So they have probes that are specific for E6, 7. So it's one RNA with two open reading frames. They have a probe that will hybridize only to the E6 and 7 from strain 18. They have another probe that will hybridize to the E6 and 7 that's only from strain 16. And so in the presence of E6 that is not under the, this is so clear in my head. E2,
E2.
when expressed, shuts down transcription. So it's the E6 and 7 under control of the viral promoter are shut down by E2. The E6 and 7 that are provided on another, they're under an SV40 promoter. So they're made regardless of whether E2 is made or not. That's the point of this video.
Wait, say that one more time?
I should have started with that.
Yeah.
So, there are two sources of E6 and E7. One is the provirus HPV 18 that David Lax infected Henrietta Lacks with 60 years ago. That's it.
Okay.
So it's in the cells. It's under control of the viral regulation. The researchers at Yale have infected or transfected the cells. It's a different, so it's with a copy of the gene that's under control of a strong promoter, SV40. So it's made whether E2 is there or not. It's not under control of E2 transcriptional regulation.
So it's separate.
Yes. So in these cells, so the mock infected, E6 from HPV 18 is present. E6 from HPV 16 is also present. So here's proof of their, let me give you the bigger picture. Where they're going is to tease out what happens with E6 and what happens with E7 in the cell. And to do that, they need to eliminate the other one. So here's E6, 7 from 18, infected with E2, not there anymore. The one from 16, even when it's been infected, is still there. Why? It's not under control of that promoter. It's made from it's driven by the SV40 promoter. So that, and so this is actually a copy of 6, and this is a copy of 7. You'll see that they're a different size, because this isn't the RNA encoded by the viral genome. It's the viral gene put in a vector that produces a copy of it. So the E6 and E7 from strain 18 are always made because they're under control of the strong promoter, whereas the E6 and E7 from 18 are under control of the HPV transcription, and thus you can shut them down by providing E2 to the cell. Everybody with me? All right, HPV. Yes.
So, theoretically, if E2 were present in the cells of Henrietta Lack, then she would not have the persistent HPV 18 infection.
Yes.
Okay.
So with a caveat, E2 is there. It's just not, so, slightly harmful. I don't know.
Yeah, other cells.
Look at figure 2.
All other cells.
Sorry, let me come back to this. So, I'm sure E2 is regulated also by the virus. It doesn't want to turn, so let me go back to why is it named E? Early. So we've seen that before, naming viral genes. We have early and late genes. We get to the late genes in the viral replication, you do want to shut down 6 and 7. But they're not normally, it's clearly they're expressed in the HeLa cells as things go. It's when this copy that's provided is also under control of a different virus.
Okay.
Strong, they're actually infecting the cells with a virus that produces a lot of E2.
Right.
So they're making the situation artificial by giving more E2 than would otherwise be there.
Okay.
Clearly there's not enough there to inhibit the tumor suppression pathways, or she wouldn't have had cancer. If you read the paper, you'll notice that's the last thing the authors say, is that because endogenous senescence or apoptosis, that's going to be the difference in pathways, are activated if expression of either 6 or 7 is repressed, therapies directed against either viral protein may be beneficial. So it would be a target for gene therapy to introduce a virus that expressed E2 and shut down the suppression of the tumor suppressor pathways. So it's in theory. We're understanding the biology of the virus here. So let's go to a more simple example. By the way, the chapter I'm gonna cover on Monday is 4 pages long or something like that, so Brad will just carry this discussion over, because at the very least, I'm having a good time. All right, see, here are my notes for this one, this is very easy.
You're having too much fun with this one.
This is what, so when you become a scientist, this is what you do, right? You run gels and you interpret them. Let's do it.
So how many papers will we be responsible for the final?
So far, 2. What do we have, 2 more weeks? I'll do it, I won't give you a paper the last week, so. Yeah, read 2, 3, 4. I like this one a lot. I did look at this on Monday and went, oh, this is a lot to ask of them. And it is, but one of the reasons why I'm talking about it is to hopefully explain it on a way that, one, you might learn something, 2, you get a taste of viruses and cancer. And this example. So here we are. So this is now not a northern blot, but a western blot. They're looking at protein levels. So P53, and this is the PRB, or P105, or as they now say, 105RB. This, by the way, in many respects, is a very poorly written paper, because they don't, for example, tell you that 1 and 4 are code numbers. You just kind of have to figure that out. And they don't tell you what LXSN means. It's just the name of it, right? So,
And LXSN is the control.
yeah, it's the empty vector for these 2 G's. So this is expression of nothing. This is expression of E6, this is expression of E7. Let's start up here. So there's 2 tones of each, they're always all the same thing, so just pay attention to one. Reduce your world to that box. Or actually, let's reduce your world to this box. Once again, minus is infection with the empty virus, plus is infection with E2. Interpret this.
If
It's
E2
expressing
suppresses the E6, then the E6 can't degrade the P53.
And thus P53 is there. That's hard. Good job. So in HeLa cells, E6 and 7 are or destroying P53. If you repress the expression of E6 and E7, you get P53 back. If you now infect with E2, knocking out expression from the strain 18 E6 and 7, but then provide back a copy of E6, you don't get P53 back.
Isn't that why the HPV is 16 then?
Yes, so E6 and E7. E6 binds P53, leading to its destruction. If E6 isn't there, P53 not destroyed. So here's a wonderful example. That figure in your textbook came from somewhere. Your textbook doesn't actually reference this paper, so it didn't come from here. If E6 is present, even though E2 has eliminated expression of E6 from strain 18, that was this question. Where the strain 16 E6 is there, it's still binding P53, causing its degradation, thus it's not in the western blot.
Even with E2 and 16?
Yeah, so there, right. Oh, there's no E2 from strain 16. Right. HeLa cells are infected with strain 18.
Oh, okay.
E7, remember, binds PRB. So if we infect with E2 and knock out E7 from 18, and then provide or E6 and 7 from 18, and then provide a functional copy of 7, we get P53 back. Why? E6 is gone. So I said before, what these researchers are trying to do is tease apart the effects of E6 from E7. By George, they've done it. This was so cool, they showed me, like, this is amazing. The P RV story is a little more complicated. It has a phosphorylated or unphosphorylated, it's actually phosphorylated, more phosphorylated, but you can imagine addition of a phosphate to a protein or addition of multiple phosphates to a protein and 2 negative charges per phosphate is going to alter the mobility of a protein in SDS. So this is the active form of P105. This is the inactive form of P105. So, go back to the control. Now we're coming down to here, we'll come back to these. In non-infected virus, we have active P105. When we provide E2, I'm sorry, uninfected is kind of a misnomer here, everything's infected. When we infect with the E2, we suppress 6 and 7, that leads to an accumulation of the non-active RB.
And how is that determined, how can you determine that just by looking at that band?
Almost, so previously known
Okay.
is the answer. Somebody at some, so people who know gels are, oh, that's a phosphorylated, that's an unphosphorylated.
Okay.
But you could, somebody would have done the experiment, I'm sure they referenced it here, where you could add a phosphatase that would remove phosphates and show that this band becomes this band. That's probably what was done. You can do mass spectrometry, you can do a lot of things to actually look at the phosphate there. It's an antibody to the protein that clearly recognizes either form. So you're gonna see variations in mobility. So the E6, remember it's E7 that's going to bind to P105, we see the same pattern as the control. So we're providing E6, we still get the predominance of the unphosphorylated form because E6 or E7 is not present from the overexpressed or not under controlled virus. However, here when we eliminate E7 from the different perspective. When we eliminate E7 from the HPV 18 and provide an E7 from an exogenous source not under control of E2, when we infect, we get a destruction of this non-phosphorylated form. So it's I'm sorry, we're going to go to they have a second experiment to explain this, but let's suffice it to say E7 is still doing the job of binding P105, leading to its inactivity. But not P53. Yeah, simple figure here. really quite simple. Actually, it's, let me come back here. So, P21 inhibits CDK2, so it's a cell cycle regulating kinase. So P21 and then later cyclin A are both downstream targets of the tumor suppressor. So if P21 is present, P53 is working. So they're showing here that the tumor suppressor pathway, not just the tumor suppressor protein, are turned on. Sorry. Yes, so P21, so we have cell or cyclin-dependent kinase 2. So the protein's called cyclins that are expressed at various points in the cell cycle that control the cell cycle. Cyclin-dependent kinase 2, being active, moves the cell from G to S. So growth synthesis. P21 inhibits CDK2. P53 activates P21. I know it's really, I don't know this stuff either. But take home is if P21 is there, the P53 tumor suppressor pathway is working. And as you can see, when P53 is not degraded because E6 isn't there, P21 is. Thus, the P53 tumor suppressor pathway that is suppressed by E6 is not suppressed, but is in fact suppressing the tumor pathway. Yeah?
Like, you can't have P21 without P53.
P21 is what inhibits.
Okay. So here's how we control the cell cycle. Easy. Let's say you want to grow. How do you know you want to grow? Maybe you receive a signal like epidermal growth factor. Epidermal growth factor is an extracellular hormone that tells the cell to divide. So it's bound by a receptor. The receptor, it's receptor tyrosine kinase, like you've seen a million times. Phosphorylates itself, sets off a signaling pathway. There's protein kinase C, nuclear factor, kappa beta, yada, yada, yada. Genes turned on. Those genes turning on lead to cell proliferation. So this is kind of a bad figure in that these are all pathways, but then this is the cell dividing for some reason is inside the nucleus. Well, but you need to replicate the DNA, for example. So there is nuclear transport of these things, and then genes are turned on, and those genes turning on lead to repression of cell proliferation. This arrow should be this way, by the way. Here it is, P53. In this, it's activated via the ARF pathway. But P53, nuclear protein, it's going to activate P21. It's going to inhibit CDK2, which leads to cell proliferation. So if you inhibit CDK2, you arrest cell proliferation. Your tumor becomes non-tumor. And it's because you activated P21 because you had P53. In the virus, we've lost the P53 because E6 is targeting for destruction. We lose active P21. We get CDK2 activity, therefore we get cell proliferation. Like I said, simple. Or you've de-suppressed the tumor suppression, thus suppressing the tumor. You wonder why I'm a bacteriologist. You know what? I really like this. I hope you guys are doing it too. You're at least not the word senseless, that's a good thing for most of you. Pick it up on Monday. Go read the paper if you haven't.